IonQ is here, so it's quite active right now. Not to mention IBMQ has inside the NC State. So again, we started about 2 years ago. When we started, we worked with Fujitsu on their digital annealer, but since then we've started to work with D-Wave on their quantum annealer. So what do we do in Polaris? We are offering rapid drug discovery. So we are designing new molecular drugs. We are doing it both as a fee for service and designing our own assets. And we're doing this using quantum computing. And the power of quantum computing that we are using is the optimization tool. So we are building very large, vast chemical space, virtual chemical space on the computer, and we are using the optimization tools inside quantum annealing to find the best molecules inside that very large chemical space. So why? Okay, the first thing, and why us and why now? We have the first drug discovery platform that is built on a quantum computer. I think we also have probably the only drug discovery platform that is built on a quantum computer. And we are again using it to find the right molecule, the molecule that will become the best drug in a very large chemical space, in a virtual chemical space. So I don't know how many of this team know the process of drug design, drug discovery, but what happened with drug discovery is that for a certain disease, you have a certain biological target, a protein, and many drugs stop the activity of that protein. They block, they inhibit the activity of that specific protein, and that way they stop the disease progression. So we're looking for a small molecule that will bind into a very large protein and stop its activity. So we're looking for something that will fit inside a very specific pocket. Now, for these molecules, you can design them on the computer. This is something that the technology is available. The industry knows how to do it, but the industry knows how to do it on very small scale, a couple of thousands of molecules at the most. And what we are saying is, instead of doing it a couple of thousands at a time, design a much larger chemical space, work with billions of molecules, and find the molecules that you are looking for the first time around. We are finding molecules among billions, and we're finding as good molecules as the industry or better. We're doing it way faster, that's for sure. We are using the D-Wave digital quantum annealer. And in order for us, when we search inside our chemical space, which is again, billions of molecules, it takes us less than a minute to scan that large chemical space. When we are doing it, we can do it brute force. So we can run it, for example, on Google. This will cost us between 1,000 to 10,000 times more. So we can maybe do it for 1 billion molecules, but when we do that on 10 billion molecules, this is becoming untenable. That's why we're using a quantum computer. I'm not going to go that much more into the technical aspects of it. So if anybody has questions, comments, I'll be happy to answer those.
Yeah, so obviously we have a number of technical people in the room here. I guess starting off, so my question is, what types of approximations do you have to make before quantum hardware gets super duper awesome?
So molecules are three-dimensional objects, okay? And what we're doing, and you can see the funnel here, what we're doing is we are oversimplifying the shape of the molecules and getting it more and more elaborate. So when we describe the molecules, which are three-dimensionals, but when we're looking at them inside a quantum computer, we're doing it as a two-dimensional object. Okay, so we are first doing a two-dimensional scan, and then we're doing a three-dimensional scan, and then the interactions of three-dimensional objects. When we're doing a two-dimensional scan, that's when we are using the quantum computer. And we are looking at molecules that can become good drugs. That's what we are doing there. We're looking at billions of molecules and finding the best ones. These are the ones that will go to a three-dimensional description. And we start from billions of molecules that are two-dimensional. We choose from those about 100,000, which are the best ones that will go to the three-dimensional descriptions, because we cannot do more than 100,000 molecules for three-dimensional, sadly. When one day, when gate computers will be more useful, then we will be able to run three-dimensional also on a quantum computer.
Very cool. So I'm assuming a lot of these are hybrids, so they have classical components, you know, CPUs mixed in with the QPUs. Is that what you're mostly leaning on? Is like, say, for instance, the hybrid quantum annealers for D-Wave?
Yeah, we can use the hybrid quantum annealers. We don't need to. Their system is large enough for us. We are using a bit below 1,000 qubits, so we can run on their current advantage as is. We are using some of their newer algorithms that we find extremely, some of their solvers are extremely useful and very fast.
Gotcha. So Philip, I said you popped on. Anybody else with questions for Shahar right now regarding to the presentation, drug discovery? And yeah,
yes.
go ahead, Tim.
Yeah, I was just gonna ask a question. First of all, I'm gonna say that getting my PhD in physics, I did molecular dynamics simulations on proteins, and the proteins were the ones that cause Alzheimer's disease or other neurological diseases. So anyways, and then the hypothesis for my dissertation was that we have these types of proteins, they're like in a hairpin shape. Right, and they have to stretch out in order to aggregate, and then once they start to aggregate, they become damaging. And so I came up with the idea of these things called alpha bodies. They're called alpha bodies, which are not well known. So anyways, the idea is we could use like these alpha bodies to clamp down on these proteins to keep them in the hairpin shape, which can stop them from straightening out and then aggregating and then becoming damaging. So I wonder if, you know, you could do that kind of stuff, or we'd be interested in that kind of thing in your company.
So I don't know specifically about Alzheimer.
Okay.
I really am not familiar with that, but there are several other diseases where the aggregations of proteins is a problem. Okay.